My name is Monica Wesley, and welcome to Ask the Expert. This is a brief, informative, and lively discussion with experts in the type 1 diabetes and related interdisciplinary research fields. We're recording this event. We're going to post it on the Sugar Science after the event, and it'll also be housed on our YouTube channel. And if you have questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. And today we have two guests, very lucky to have two guests. We have Carmen Valcarce, she's PhD. She earned her doctorate in biochemistry and molecular biology at Universidad Autónoma de Madrid in Spain. She was a project leader on the Glucokinase activator project at Novo Nordisk, and she's currently CSO at vTv Therapeutics based in North Carolina. She is a pharmaceutical executive with 20 years plus years of research and development and experience in life sciences, including preclinical and clinical development in the fields of metabolism, diabetes, and Alzheimer's disease. Clara Klein is MD PhD. She was born and grew up in Atlanta. Then she traveled cross country to do her undergraduate work at Stanford University. And this is very interesting. She pursued research in synthetic organic chemistry, just one of the more easier subjects. She then joined UNC's MD PhD program and completed her PhD in cell and molecular physiology under the mentorship of Dr. Kathleen Caron. Her research focused on the role of a small peptide hormone, which is adrenomedulin, in the development of lymphatic vascular system, an understudied vessel network, very true, which increasingly is recognized to play really critical roles in normal physiology and disease states. So using in vivo and in vitro techniques, she helped identify a new role for G protein coupled receptors, GPCRs, and particularly decoy receptors in lymphatic vascular development and function. It's really interesting work. She then entered the research pathway training in internal medicine and endocrinology, and she has plans to pursue a career in translational medicine, focusing on multidisciplinary approaches to diabetes and obesity. And so she hopes to bridge the gaps between basic and clinical research in order to address the ongoing epidemics of diabetes and obesity. So welcome, both of you, Clara and Carmen. Thank you very much for taking time out of your busy schedules to speak with us. And today we're going to talk a little bit about TTP399 and also the Simplicity T1D trial. So TTP, the tongue twister, 399, is a novel hepatoselective glucokinase activator, and it's in development for the reduction of hypoglycemic events in patients with T1D. And the Simplicity T1D trial is a study that showed that treatment with 800 milligrams of this TTP399 demonstrated statistically significant reductions in HbA1c and clinically relevant reduction in the frequency of severe hypoglycemia, which is always, something that is in everyone's mind to be avoided in the type 1 diabetes world. So welcome, and please, let's launch into this presentation.
Sure. Thank you. Thank you so much for having us. Like we just heard, my name is Clara Klein. I'm an endocrinology fellow and a member of the physician scientist training program at UNC Chapel Hill. And I'm really thrilled and lucky to be part of the team that's helping develop TTP399. And I'm delighted to be here today with Carmen to talk to you about hepatoselective glucokinase activation for the treatment of type 1 diabetes. Some of the data that I'll present today were previously published in the April 2021 edition of Diabetes Care. And so while I will talk about how we think this drug works and what we've previously shown, I'll try and highlight why we're so excited about the potential of glucokinase activation and TTP399 in particular for the treatment of type 1 diabetes. I have to admit that as a young endocrinologist and a clinician scientist, I found it somewhat stunning that despite how much knowledge we've gained and how much technological advances we've made, there's still so much to learn and so much to do to improve the care of our patients with type 1 diabetes. Despite truly revolutionary therapies for the treatment of type 2 diabetes, treatment for type 1 diabetes remains almost universally insulin alone. And in the United States, there are no FDA-approved oral adjunctive therapies for the treatment of type 1 diabetes. And despite more physiologic and faster-acting insulins, very few patients are achieving the ADA-recommended hemoglobin A1C targets of less than 7%. And the truth is that they're not even getting very close. And this is particularly true for our youngest patients who will live with diabetes the longest and are therefore at risk for the most severe complications. But maybe even more interesting, despite all of these advances, some of the more severe and acute and preventable complications of type 1 diabetes, including diabetic ketoacidosis and hypoglycemia, are increasing. And so you can see here in the graph in the middle that there's been a sharp increase in the incidence of diabetic ketoacidosis hospitalizations in the last decade. And that's also true for hypoglycemia. That may be a little bit more obvious here in this figure, where you can see that the annual prevalence of severe hypoglycemia in patients with type 1 diabetes has increased substantially over the last decade. So much so that the Endocrine Society, among other drug agencies, are identifying reducing hypoglycemia as a strategic priority and really a primary area of concern. And yet, despite all of this interest, there are no therapies available that protect against these events. To date, most, if not all, of the adjunctive therapies tested in patients with type 1 diabetes may improve glycemic control, but they do it at the expense of increased hypoglycemia and ketosis. For example, the SGLT2 inhibitors or the GLP-1 receptor agonists when tested in patients with type 1 diabetes. So there remains a very clear unmet need to identify therapies adjunctive to insulin that improve glycemia to prevent those long-term microvascular events, our poor outcomes, and also to without increasing these acute severe events like ketosis or hypoglycemia. But if we get a little bit greedy, if we move a step further, if we could identify an adjunctive therapy that protects against these complications while improving glycemia, it would mark a very significant advance for the treatment of diabetes. And so what I hope to convey to you today is that we see real potential for glucokinase activation for the treatment of type 1 diabetes, not only to improve glycemic control, but to provide some protection against hypoglycemia and ketosis. So how does TTP-399 work? So TTP-399 works on glucokinase, and glucokinase is the enzyme that catalyzes the first step of glucose metabolism or glycolysis in both the liver and the pancreas. Glucokinase has unique properties that allow it to act as a molecular glucose sensor. Specifically, the kinetics of glucose binding is different than for other hexokinases, such that it binds glucose at a much higher concentration. And that glucose binds glucose at a much higher concentration is important because in the pancreas, glucokinase activity leads to insulin secretion. So glucokinase activity really should only occur during periods of hyperglycemia. A second key property is that glucokinase is not inhibited by downstream products like glucose 6-phosphate, such that it continues to be active as long as glucose concentrations are high enough. Again, insulin should be secreted during periods of hyperglycemia and should only stop when that hyperglycemia has resolved. There is also strong genetic evidence that supports glucokinase's central role in glucose homeostasis, as both activating and inactivating mutations have been described that lead to dysglycemia. Particularly relevant to our discussion, inactivating mutations in glucokinase lead to heritable forms of diabetes like MODY2. And so it's not terribly surprising that glucokinase has long been a therapeutic target of interest for the treatment of diabetes. But development of glucokinase activators has been stymied, as you can imagine, because of unfettered activation of glucokinase within the pancreas, which leads to insulin hypersecretion and hypoglycemia. And as we've discussed, hypoglycemia is one of the major problems of adjunctive therapies, particularly in patients with type 1 diabetes. Hepatic glucokinase, however, has several properties that differentiate it from pancreatic glucokinase. First, hepatic glucokinase has a unique interaction with glucokinase regulatory protein, or GKRP. During periods of euglycemia, GKRP binds glucokinase and sequesters it within the nucleus. During periods of hyperglycemia, glucose binds glucokinase, and glucokinase undergoes a conformational change, which dissociates from GKRP, and GK moves into the cytosol and allows glucose metabolism through glycolysis. Secondly, whereas pancreatic glucokinase is constitutively active, glucokinase expression in the liver is insulin dependent. So in type 1 diabetes, where portal vein concentrations or the portohepatic insulin gradient is low, it's expected that GK expression and GK activity is also low. And so if we can selectively restore glucokinase within the liver in people with insulin deficiency, we may have a unique opportunity to improve glucose homeostasis without leading to insulin hypersecretion and hypoglycemia. So TTP-399 is a small molecule glucokinase activator that activates glucokinase within the liver, but has no activity within the beta cells of the pancreas. Importantly, TTP-399 does not interrupt the GK and GKRP interaction, and so it maintains physiologic control of glucokinase and only promotes activation during periods of hyperglycemia. TTP-399 has been tested in patients with type 2 diabetes and demonstrated to have a favorable safety profile without hypoglycemia and a significant improvement in glycemic control over a period of 6 months. Prior to Simplicity T1, no glucokinase activator had been tested in people with type 1 diabetes. So we don't need to spend too much time talking about the study outline for Simplicity T1, but I do think that it's really important to understand that Simplicity T1 followed an aggressive treat to target design, such that patients that were randomized had to achieve a target of 80 to 130 milligrams per deciliter fasting prior to randomization to TTP-399 or placebo. During the study, there was insulin adjustments frequently to target, again, a fasting plasma glucose of 80 to 130 and a post-meal plasma glucose of less than 180 and a peak of 200 mg per deciliter. So this is very tight control. And this was targeted in both arms, and it was blinded. And so any improvements in glycemia above placebo was seen on top of this intensive treat to target adjustments in insulin. So as we talked about, these data have been published in Diabetes Care. And so I'll just briefly run through the key study findings and then talk about the things that we're looking to in the future. First, there was a statistically significant reduction in hemoglobin A1C, again, on top of this aggressive treat to target design. And this was also represented in a 2-hour increase in time and range relative to placebo in patients on TTP-399. Total daily insulin dose was also numerically reduced relative to baseline. And I think that this piece of data is some of the most convincing that we have. So I'll just kind of walk you through this figure. So this is a plot of change in insulin against change in total insulin. And so if you go on the y-axis, it says change in hemoglobin A1C. And you can see that if you go down, you have improvements in hemoglobin A1C, they're going down. And if you increase hemoglobin A1C, glycemia is getting worse. And the same is for on the x-axis is insulin. If insulin is decreased, you go to the left. And if insulin is increased, you go to the right. And so in placebo, patients randomized to placebo, and these are these orange circles here, you can see what you would expect. If you reduce insulin, so insulin goes down, glycemia gets worse, hemoglobin A1C goes up. If you increase insulin, the glycemia gets better, and you have a decrease in hemoglobin A1C. In this quadrant here, where we're reducing insulin and we're also seeing improvements in glycemia, this is dominated by these blue triangles, which are the patients that were randomized to TTP-399. So they're having improvements in glycemia even though their insulin is going down. Again, really highlighting that we think that TTP-399, and the evidence shows that TTP-399 is improving glycemia and allowing patients to reduce their insulin doses. More importantly, there were no detrimental safety signals that were seen across multiple parameters. And this really stands in distinct contrast to other agents that have been tested in type 1 diabetes, including SGLT2 inhibitors and GLP-1 receptor agonists. So like I said, overall, the treatment emergent adverse events were similar between the groups, but we did note a numerical reduction in hypoglycemia and ketosis. And this is kind of the start of one of the reasons why we're so excited about these agents. Post-hoc analysis revealed a statistically significant 40% reduction in hypoglycemia events in patients randomized to TTP-399, and that's represented here. Ketosis events, again, defined by a beta-hydroxybutyrate greater than 0.4 millimoles per liter at any study visit, were overall few. And the study really, again, there were few events and it was not powered to assess statistical changes. And so we didn't perform statistical analysis. But I think you can appreciate that there was a reduction in ketosis events in patients randomized to TTP-399 compared to placebo. And so this was a very encouraging result. So those of you who are paying very close attention may wonder, you highlighted that this was an intensive treat to target design, and maybe people who are having less ketosis were just on more insulin, and maybe people who are having less hypoglycemia were just on less insulin. Maybe the change in insulin and that aggressive treat to target really impacts these results. But when we analyze based on change in insulin, the data really don't show that. In fact, they show that regardless of insulin status, participants on TTB-399 experience less ketosis. So whether they reduced their insulin, they had less ketosis, whether they had stable insulin or they increased their insulin. And that's really also true for hypoglycemia. As you can see, at every, whether they reduced or had stable insulin, they had lower amounts of hypoglycemic events. In the increased insulin group, you can see that there was one participant who had symptomatic hypoglycemia, but this patient had no detectable TTB-399 at their study visits. So I think, again, this is really encouraging data, but when I look at this again, this is just a representation, another representation of that cross that I showed you earlier. What I think is incredibly impressive when you look at it another way is that in participants randomized to TTB-399 who reduced their insulin, so 15 participants reduced their insulin, 67% of them also improved their hemoglobin A1C. And despite having less insulin on board and better glycemia, they had less ketosis events and less hypoglycemia. So this is, I think, really encouraging data, and it's consistent with preclinical data and animal models done by Carmen and by other groups that suggest that glucokinase activation, restoring glucokinase activity, really can have an impact on ketosis and hypoglycemia. So we're moving on to test this hypothesis in humans, and we're currently conducting a phase 1 mechanistic study to determine the impact of TTB-399 on ketone levels during insulin withdrawal. Again, the study is outlined here, and briefly, we're randomizing patients to TTB-399 or placebo. It's blinded for 7 to 10 days, and then they come into our clinic and we turn off their insulin pumps for 10 hours and measure ketones. And we are in the midst of it, so we don't know what's happening because it's all blinded, but we're really interested to see if it's possible that TTB-399 will delay ketone rise in humans. But maybe even more importantly, there's a pivotal trial that's coming. The FDA has given TTB-399 a breakthrough therapy designation, and that's primarily based on its impact on hypoglycemia. Because as you mentioned at the beginning of this talk, hypoglycemia is a major issue. And so if we could address hypoglycemia, it would have a really important impact for our patients. For that reason, it's expected that hypoglycemia will play a more central role in the trial than it has in other adjunctive therapy trials. And we're still working on the details for how this trial will be designed, but I think that it'll be really interesting. The phase 3 trial will be really interesting, not only because we are encouraged and excited about the drug, but also this will be an interesting and I think novel type of trial design for adjunctive therapies. So in summary, the Simplicity-1 study confirmed the efficacy and safety of TTB-399, a glucokinase, hepatoselective glucokinase activator in participants with type 1 diabetes. But really one step further, we saw a favorable safety profile and maybe even a role for protection against hypoglycemia and ketosis events. While longer and larger clinical trials are needed and currently being planned, we're really encouraged about the future of glucokinase activation in patients with type 1 diabetes. Really, not only because it gives us more therapies available for people with type 1 diabetes, but really primarily because it will improve patient quality of life. So the patients that I see in clinic every day, our patients are constantly struggling with the push and pull between too much insulin and hypoglycemia. They do this every day. That's something that they deal with every day. And so if we could find a therapy that gives some peace of mind with regard to severe hypoglycemia or ketosis, it would really not only be a major advance for the field, but individually would provide significant relief to many of our patients. And so we're really excited about the potential of TTB-399 for our patients with type 1 diabetes. So with that, we're happy to take questions. And I guess I should just take a moment to say that none of these studies can be done without the incredibly generous spirit of our patient population with type 1 diabetes who participate in these trials, and the hard work of our study staff. So always want to give a shout out to the hard work of our patients and staff. So happy to take any questions.
Yeah, that's fantastic. This is really encouraging data. And just I have a few questions, and I'm hoping people will jump in. I wondered, what sort of go back to the very beginning of your presentation, like what is behind this sort of real increase in severe hypos, increase of DKAs and HbA1c? Is there any, feeling in the clinic, like what is driving these surges?
Yeah, I don't think that we really know. I think it would be interesting to, so some people say, okay, there's just more people with type 1 diabetes being diagnosed with type 1 diabetes. And I think that, also with the advent of all these technologies, I do think that with more people on insulin pump therapy, when the insulin pumps fail, people are at more risk for diabetic ketoacidosis. Certainly, the advent of these therapies that people are starting to use, kind of even independent of FDA approval for type 1 diabetes, like the SGLT2 inhibitors, put people at risk for DKA. But I do think it's more complicated than that. And I don't think we know the entire answer of why DKA is going up, but we're certainly seeing an increase. The hypoglycemia, again, as we start to target, again, we're targeting fairly tight control. With the guidelines are a hemoglobin A1C of less than 7%. I think there are a certain population of people that, if you're trying to target that, then you're just going to have hypoglycemia, because it's hard to get there without having times where you have too much insulin on board. And so again, if we're going to achieve, we haven't found any data that exists that says that normalizing blood sugars completely in patients with type 1 diabetes improves outcomes. But we've never had anything that allows us to get there without hypoglycemia. And so I don't, these new technologies and the advent of new therapies like TTP-399 may allow us to see, do we reduce macrovascular outcomes by getting people to a hemoglobin A1C of 5.8% without hypoglycemia?
Yeah, no, it sounds like the TTP-399 sort of rounds out the profile on both ends, right? I mean, you're sort of rounding out the excursions on both ends. So, which is really pretty impressive. I mean, if it can really pull through to a larger trial, it's really going to be a great tool in the toolbox for type 1 diabetes. And I also wondered, what was behind the actual, how was TTP-339 originally identified? What was it, how was it, when was it identified as a target molecule for type 1 diabetes, and sort of like, what's the history of it?
Yeah, I think I probably have to defer a little bit to Carmen here. People have been developing glucokinase activators for, I think decades, early 2000s. There have been issues, including not only global activation causing hypoglycemia, but also issues with the liver profile, because of the way that it can impact, metabolism, obviously. And so what's unique, we think that this interaction, maintaining the interaction between GK and GKRP, really protects against some of these adverse events. But Carmen really will be able to speak in more depth about that, since she's been, this is her baby.
Yes, as Clara said, we started many years ago in the early 2000s, when everybody was working on glucokinase. We had a different approach. We, from the beginning, didn't want to activate glucokinase in the pancreas, in the beta cell, because of the people that have mutations that activate glucokinase overall. they suffer of hypoglycemia. So we thought that we will not be able to control that. And so we were looking for compounds that only activate glucokinase in the liver, that we thought that that was the only way that it could be safe to activate glucokinase. So we took some time to get the right molecule, but finally we got it. And because of all the effect is coming from the liver, we thought that this could be very useful in people with type 1 diabetes, because you don't require, it's not a compound that is increasing insulin secretion. It's a compound that is increasing the metabolism of glucose in the liver. So that's why we decided to go this path and try this compound for type 1 diabetes.
Yeah, that's great. I just was curious about the history to trial, what, how long and so forth. So that's really interesting. And we have a question. Is vTv Therapeutics specifically searching for a European partner, given that they've been attending EASD conferences in recent years? And will they attend the upcoming conference, September 27th to October 1st?
We definitely will need partners. So we are open, to European partners for sure. Me coming from Spain, I would like to have a European partner, and very probable we will be attending the ESD this year as well. Yes. Great.
Yeah, no, I think it is. We've also tried to focus on scientists and small startups, entrepreneurial startups, biotech that is outside of the US. We really are very interested in sort of gaining a global audience, as well as sort of highlighting the global effort to work on type 1 diabetes. So I did also wonder, like, when you are putting together your phase 3 trial design, are you thinking about, you're talking about people who are already in tight control. You're thinking about maybe targeting athletes. I know that's a lot of people are looking at athletes right now in that bandwidth, or is it going to be more of a larger, a larger population you're kind of looking to draw in?
Yeah, I think, it's, picking the right population is always a challenge.
Yeah.
I don't think, again, Carmen will jump in, but I think that this drug, my hope is that this drug will be impactful for everyone, and the great thing about this, and I didn't spend a lot of time talking about it, is it really seems to impact prandial blood sugars. It seems to be most effective on bolus insulin. And so
that's good.
The people that I worry about the most are the people who just cannot manage to give themselves insulin for their meals. And so if people who have hemoglobin A1C of 10%, we haven't really seen the patient population with that. We haven't tested the drug in patients who have very poor control. The average in Simplicity T1 hemoglobin A1C average was 7.6%. It's very good control, even though we're always targeting better. Who knows what it would do for a patient with a hemoglobin A1C of 10%. So, again, the study details are to be determined, but I do not expect that we're going to restrict it to people who have very good control.
Okay, that's great. And here's another question. The longer and larger clinical trials will be costly. Do you think a partner for TTP 399 could help fund the trials?
I think that's maybe a Carmen question, not a me question. I think I'll leave that one to Carmen. She may not have maybe she'll come back.
Well, there is no doubt that it will be costly. We have the finance now to do it ourselves, but we are always open to partnerships, because many times you get more from working together with others. So if the right partner come across, we will definitely entertain that. But as it is now, we will continue ourselves and do the bigger studies.
Yeah, it's great to have you both on here, both from the science and the business, disciplines or backgrounds, I guess. And so I also wanted to ask you, when you design your trial, do you think of entities like T1D Exchange and now in Canada, Connect T1D to sort of, gain participants? I mean, they have a really, especially T1D Exchange has especially great record for really getting trial participants up and running quickly, or, gathering those people.
Yeah, I think, as you see, I think we, as Carmen is saying, I think partnering with all of these people can be beneficial. And so, recruitment is always the hardest part of trials. So anyone we can involve to recruit successfully, we will. So, I don't know what the plans are, but certainly we'll look into all of the avenues to get, to reach recruitment. And every site is a little bit different, obviously.
Yeah, for sure.
And we have been using some of the sites of the T1 Exchange for our simplicity studies. So we definitely are in contact with many of the sites in the network, and we plan to keep involving them for this trial as well.
Yeah, that's great. They're a great repository and connector, T1D Exchange. So yeah, I mean, I think that this is, one more question. What are the expected clinical endpoints for, I'll just take this last one, for TTP 399 mechanistic studies, and will these results be presented at the ADA conference in late June?
So I assume this is about the mechanistic study that we're doing right now. So the study won't be completed by the time ADA rolls around. So the study endpoints are currently our increase of ketones by a certain percent, and or an absolute amount. So the data is coming, and I think that in my preliminary, I don't know who's on what, but I think that there's going to be a huge amount of data to mine. So there'll be, I'm really excited to see what we learn from this. But I think, there will be lots of interesting secondary outcomes that we'll have to look at. The primary outcome, again, is just a rise in ketones.
Yeah. Well, the end of June is just like 3 weeks away. So we've had fast time.
Here at UNC, we have done, we've done a number of, there's 2 sites in this study, and we've done a number of insulin withdrawal tests here in Chapel Hill. And it's been really interesting.
Yeah, well, we cannot wait to see what happens next with vTv. And we thank you both for taking time out of your busy schedules to meet with us. Carmen Valcarce and Clara Klein, thank you again. And really exciting to see what's next for vTv. Thank you again.
Thank you so much for having us. Really appreciate it.
Thank you. Thank you, Monica, for inviting us.
Thank you. We wish you all the best.